A big hello to all my future doctors out there. I hope my dear children are doing really great, and I hope you are watching the videos that we are trying to make for you. Well, if you're not, then definitely you should. If you're not able to watch the entire video, then what you can do is pause, okay, and at least see the questions which are on there. It will be really beneficial for you. Okay, so it's just previous year questions that I have made you practice over here. So those of you there with me might be knowing that, okay, I try to make you understand certain concepts also together with the questions. So any question coming off from the same concept, you would be able to solve that as well. So that is the use of watching the video, or if you just want to see the question and the solution, you can just roll down with the help of the slides out there. Yeah, now my dear children, also one more thing, if you have not started attempting mock tests, please make sure you are doing it on time, because you just have few days left. Yeah, almost I'll say 5 to 6 days are left, and you should be definitely giving 1 or 3 mock tests for sure. Okay, with that limited amount of time that you have to see whether you are able to solve questions. Okay, so sometimes we have the knowledge and all, but we won't be able to put that knowledge with the amount of time that we are given. So that's a very important practice that you should be having. And if you have not found a good mock test, we are providing you free mock test. You can find the link in the description box. You can click there, go to the link, okay, register yourself and then get the free access if you want. So if you do not want, if you have already practiced some other mock test, it's totally fine, but I'll recommend you definitely practice some mock test for sure. And I'm really glad, I want to tell, some students are putting down the answers in the comment section. I asked certain question in mid of the session and I got the answers. I'm really happy. Thank you so much for doing it. It encourages me a lot. Yes. So yes, let's get ahead. Sorry for taking up 1 minute of your time. Yeah. Morphology of flowering plants. We have the first question right over here. That is, the roots that originate from the base of the stem are fibrous roots, primary roots, prop roots and lateral roots. So you're talking about the roots that originate from the base of the stem. Okay, you're not asked about the radicle or things like that. You're asked from the base of the stem. What type of roots are being asked? So it's there, and the entire line is there in your NCERT. So the answer for this question is actually fibrous roots. So you have the stem and then you have the fibrous roots coming off like this. Okay, this is a feature of monocots. This is a feature of monocots, my dear children. Monocots are the one that have fibrous roots. Works? Chalo. So in monocotyledons plants, the primary root is short-lived. Okay, the primary root, they also have a primary root, but actually that is kind of short-lived, and it is replaced by a large number of roots. These roots originate from the base of the stem and constitutes the fibrous root system as seen in the wheat plant. Wheat plant is what? A monocot. That is what I was talking about. So your answer for this question would be this. Okay, lateral roots, as in you're talking about, for example, you have a primary root onto which you have certain other roots coming off, that is known as lateral roots. Prop root is somewhere that comes off from the stem, okay, at the top. That are prop roots, not in the lower position, that is within the root, within the soil. Okay, that is the difference between the normal root and the prop roots. So I hope I made you understand that. Yeah. Now going ahead with the next question. Sweet potato is a modified what? So I hope most of you might be knowing that it is a modified root for sure, because you might have studied in the chapter evolution that sweet potato is modified root. But whereas when talking about potato is what? A modified shoot, a stem, right? So over here, sweet potato is a modified what? Stem? No. Taproot? No, my dear children. Taproot and all, for example, you have carrot and all, that is a modified taproot, which is why the carrot shape look like that. Even radish, okay. Rhizome, no. What is your answer? What are you left with? Adventitious root. This is what sweet potato is. Sweet potato is a modified adventitious root that you get. Okay, always remember that. So B is your answer. Going ahead, how many plants among China rose, ocimum, sunflower, mustard, alstonia, guava, calotropis and nerium, that is oleander, have opposite phyllotaxy? Opposite phyllotaxy as in, for example, the leaf positioning would be something, would be something like this. This is known as opposite phyllotaxy. They would be opposite to each other. Then there is alternate phyllotaxy, which looks like this. One over here, another over here, another over here. Okay, and then there is vold phyllotaxy, where basically it will be like a vole. Okay, it will be like a vole. So you have 3 different types of phyllotaxy. You are asked about which among them would be having opposite phyllotaxy. That is this kind of thing. So calotropis and guava is there in your NCERT. Yeah, Calotropis and guava are there in your NCERT. Apart from that, Ocimum is also something that shows opposite phyllotaxy. So I'll say 3 of them are there. Okay, what about sunflower mustard, ma'am? They have alternate phyllotaxy. What about Alstonia and Nerium, ma'am? So Alstonia again given in your NCERT, it has Wold phyllotaxy. Along with that, Nerium also has Wold phyllotaxy. So what are we left with? We are left with 3, that is Ocimum, guava and Calotropis, each of them having what? Opposite phyllotaxy. Works? So your answer is 3. Your answer would be 3. Going ahead. In a cymose inflorescence, the main axis, have you seen cymose inflorescence? What happens is that at the end, at the tip, no, there would be it would be terminalized with the flower. They will be also having lateral, branches over there, and they also get terminalized with the flowers up there. That is what you mean by cymose inflorescence. Racemose inflorescence, as in only the lateral part would be having what flowers, and it would be the middle part which is there, it will be going up. It will be having unlimited growth up there in the case of racemose. In the case of cymose, basically it has limited growth and it is basically terminalized with the flower. So over here in cymose inflorescence, the main axis has unlimited growth. Wrong. Bears a solitary flower. Do you think it just has one flower, my dear children? No. Wrong. Has unlimited growth but lateral branches, ends and flowers. Has unlimited growth? No, Baba. It has limited growth and the lateral branches, ends and flower. That part is correct, but this is wrong. Terminates in a flower. It terminates in a flower. That is the only thing that is correct. So this is why it becomes your answer. So in cymose inflorescence, the main axis is like this. This is the main axis and it terminates in a flower. Now, the ovary is half inferior in, what do you mean by ovary is half inferior? So, in that case, my dear children, what is the thing is that, okay, suppose this is your female reproductive system that is there in the case of plants, my dear children, and just a second. That's gone. Okay, anyways, I don't want to confuse you guys. So over here, so basically it's like the the male and the female parts are almost in the same level. The ovary is not inside, is not within, it is not covered by the receptacle or things like that. It is half inferior position. It is not entirely, but in middle. It is entirely in the middle. I am not able to draw it properly. Yeah. So that is what you mean by half inferior ovary. Basically, you have to make sure that all of them are in the same line. Almost they all are in the same line. What do you call this arrangement? You call this perigynous. Okay, this is known as perigynous arrangement. And where do you find it? In brinjal, in mustard, sunflower or plum? The answer to this question is what? Plum. Perigynous ovary is seen, our half inferior ovary is seen in the case of plum. Cool? Yes. So, if gynoecium is situated in the center and the other parts of the flower are located on the rim of the thalamus, that is what I showed, okay, almost at the same level, it is known as perigynous condition. The ovary here is said to be half inferior. Example, plum, rose and peach. Plum, rose and peach, all the Libran flavors and stuff like that, no. So, plum, rose and peach are the thing that you have to remember for half inferior ovary. Fine. Now, going ahead, placentation in which ovules develop on the inner wall of the ovary or in the peripheral part. So, basically this is what it means. So, towards the peripheral part there is what? Placentation, like this. So, this is the ovule that you have and they get the nourishment from the placenta which is towards the periphery. What do you call this type of placentation as? Free central, basal, axile or parietal? Your answer is parietal. P for periphery and P for parietal. So, placentation in which ovules develop on the inner wall of the ovary or in the peripheral part, it's known as parietal placentation. Cool? Cool. Now, going ahead, match the placental types with their examples: basal, axile, parietal and free central. Choose the correct answer from the following options. So, basal, where do you think is basal? Basal is shown by sunflower, my dear children. Basal is shown by sunflower and things like that. So, A goes with 4. A goes with 4. I just have one option like that and this should be my answer. Let's get ahead. Okay, axile placentation is shown by China rose. It's shown by China rose. What about parietal placentation towards the periphery? You find it in mustard. Okay, C goes with 1. And free central, as in the placentation would be over here. So your ovule and all looks like this. Okay, this is known as free central placentation that is found in dianthus. So your answer for this question becomes C, my dear children. Okay. So this is how it looks. So this is marginal placentation that is found in pea, beans and stuff like that. Okay, and then over here you have axile placentation. This is parietal. Axile placentation as in middle it is there, yeah, but there are certain strips over here which separates it into different locules. Okay, so that is what you mean by axile placentation. You see in tomato also, you see in tomato, you see in China rose and things like that. Okay, and now going ahead, you have this is parietal placentation, towards the periphery it is there, yeah, and then this is free central placentation. In between there is no stripes up over here, right, and this is known as basal placentation. So your answer for this question becomes C. Cool? Okay. Next question. The term polyadelphous is related to what? Polyadelphous is related to androsia, or the male reproductive part, my dear children. For example, it is seen in citrus plants. Okay, what happens in polyadelphous? Basically, they would be having bunches of what? This thing, stamens. Okay, so one branch and there will be separate bunch. This is known as polyadelphous. Okay, this is known as polyadelphous. Just a second. No. This is known as diadelphous. Polyadelphous means you will be having multiple branches. Sorry. Multiple bunches, and that is what you see in citrus. Okay, there will be multiple bunches. They are all not together. So to give you a better idea, I'll make it properly. Suppose this is one bunch, then separate bunch, then next bunch, then the next bunch. Okay, this is what you see in citrus plants. Works? Polyadelphous. So this is related to the androecium. Fine. Now going ahead, the gynoecium consists of many free pistils in the flowers of gynoecium. Okay, gynoecium, basically they can be fused, all the pistils can be fused together, and that is known as syncarpous condition. Where do you see syncarpous condition? In the case of papaver. This is in your 12th grade. Okay, this is in your 12th grade NCERT in sexual reproduction in flowering plants. Now apart from that, there is also condition where the gynoecium, or the female reproductive part, they separate from each other. You call this as apocarpous condition. What do you call this? Apocarpous condition. And you say at this particular point, the gynoecium consists of many free pistils in the flowers. So your answer for this question would be Michelia. Michelia is something that shows what? Apocarpous condition. It's not aloe, it's not tomato, it's not papaver, it is Michelia that shows apocarpous condition. Talking about papaver, it shows what? Syncarpous condition. At least these two examples you should be having in your head. Yeah? Now going ahead. Which one of the following figures represent the placentation in dianthus? We just solved in the case of what, say, match the following. Dianthus shows what type of placentation? Does it show axial placentation? Does it show marginal placentation? Does it show parietal placentation? Or does it show free central placentation? The answer for this question is dianthus and primrose, my dear children, both of them. Remember, dianthus and primrose are going to show free central placentation. So this becomes your answer for the question. Okay? Cool? Next, aestivation of petals in the flowers of cotton is correctly shown in. Aestivation of petals in the flower of cotton is correctly shown in what? What type of aestivation does cotton have, my dear children? They have twisted aestivation. Twisted aestivation means one side of the petal would be above, one side would be below. That is what you mean by twisted aestivation. So as you can see, one above, one below, one above, basically one below. It will be like that. This is known as twisted aestivation, and this is what you find in cotton as well. So what is the answer for this question? D is your answer for this question. The body of ovule is fused within the funicle at what? The body of the ovule is fused within the funicle at. So basically, suppose this is the ovary, okay, and this is the ovule. Over here in the ovary you have the placenta. The placenta will be providing nourishment, and for that it has to remain attached to the ovule. Yeah, nourishment to the ovule, right? So this particular thing that will be the source of attachment, that is known as funicle. What is this thing? This is the placenta, right? And what is this point known as, where it is attaching to the ovule? It is known as hilum. It is known as hilum. Fine. So the body of the ovule is fused within the funicle, or with the funicle at what? At hilum. Okay, micropyle. What do you mean by micropyle? Micropyle is basically, so ovule which is there, it has two integuments, one integument and the second one. So there is an opening over here where there is no integument. This opening is known as micropyle. This is known as micropyle, and wherever the micropyle is there, opposite side of it will be known as the chalazal end. Will be known as the chalazal end. My dear children, this placenta can be here also. It can be anywhere. This whole structure that you see can be here also. Do not think that it will be opposite to micropyle end. No. Okay. Nucellus is what? So basically the ovule, inside that there are certain cells. Together you call these cells as what? Cells of the nucellus. Okay. Fine. So your only answer is what? Hilum. Cool. Now, which one of the following statement is correct? Seed in grasses is not endospermic. Seed in grasses. Grass is what, my dear children? They are monocot. Monocot means they have one cotyledon. They would be having one cotyledon. And apart from that, there is also one more thing about monocots, that is, there will be little endosperm that would be left. There will be little endosperm that is left. So one cotyledon is there and little endosperm is also left. Okay. So please understand, that is why they just have one cotyledon. Otherwise, usually plants have two cotyledons, that is dicots. But they are monocots for a reason, because they just have one cotyledon and rest of it is just endosperm. So it is not endospermic would be a wrong statement to say. Now mango is a parthenocarpic fruit. Do you think mango is a parthenocarpic fruit? What do you mean by parthenocarpy? Parthenocarpy means you are making a fruit without fertilization. That means, suppose this is the ovary. Here you have the ovule. Without fertilization, this ovary that you see is becoming a fruit without fertilization. So if it is happening without fertilization, this ovule which is there, it will remain like a ovule only. Can it convert into a seed? Usually it converts into a seed after fertilization, but it won't be converting into a seed if it is parthenocarpic fruits, which is why parthenocarpic fruits, my dear children, they do not have seeds. They are known as seedless fruits. Mango, does it have seed? Yes. So don't you think it is by fertilization? By fertilization? Yes. Yes, right. Usually it is by fertilization only, ma'am. And this is why this is a wrong statement. Again, you have to find what is correct. Now, a proteinaceous alluron layer is present in the maize grain. What is maize? Again, it is a monocot. Again, it is what? A monocot, my dear children. And in monocots, my dear children, the endosperm which is there, it's covered by a layer. The endosperm which is there persisting, yeah, it is covered by a layer that is known as alluron layer. And this alluron layer is very much important for the process of germination to happen, for the process of seed to germinate, yeah. So, yeah, that is correct, exactly correct. A proteinaceous alluron layer is present in the maize grain, correct. Now, a sterile pistil is called a staminoid. A sterile pistil, do you think it is known as a staminoid? Sterile stamen could be known as a staminoid. Pistil is a female part, right? So this would be again a wrong statement. What is the only correct statement that we have over here? The answer to this is C. Which one of the following statement is correct? It is C. A proteinaceous alluron layer is present in the maize grain. Fine? Cool? Next. The correct floral formula of chili is. What is the correct floral formula of chili? Chili comes under which family? Chili comes under Solanaceae family. Solanaceae family, right? Okay, and in Solanaceae family, my dear children, what is there? They are actinomorphic. Can they be cut into equal halves from any plane passing through the center? Yes, ma'am. So can I represent it by this? Yes. So all of them are like this. All, man, my dear children, in Solanaceae family, the flowers are bisexual. That means they have both male and female. So over here, all of them have it, ma'am. Okay, talking about the calyx which is there. Talking about the calyx which is there, the outermost green sepal, it is made up of sepals, right? Now, sepals which are there, they are 5 in number. Yeah, they are 5 in number, and all 5 of them are fused together. Are what? Fused together. They end to end, they are fused together. So this is fusion, which is why it is in bracket, 5 of them in fusion. 5 of them in fusion, but this is not. So I'll cut this off. This option is out now. Okay, this is also fusion. Now talking about the corolla which is there. After the calyx, you have corolla which is made up of petals. Same thing with petals also. They are 5 in number and 5 of them are fused together. 5 of them should be fused together. So here fusion is not there, so I'll cut this off. Okay, here fusion is there, here also fusion is there. Talking about the androecium. Okay, androecium, what about them? Again 5 of them, 5 stamens are there. Okay, 5 stamens are there, but are they fused? Are the stamens fused to each other? No. So over here it says that stamens are fused to each other. This is really wrong. What are we left with? This. So here the stamens are not fused, but basically you see a line like this. What does this line suggest? That this is corolla, that means it is made up of petals, my dear children. And this is what? The stamens. The stamen is attached to the petal. The stamen that you see, it is attached to the petal. This is known as, this condition which the stamen is attached to the petal, it is known as epipetalous condition. Okay, this is known as epipetalous condition. Okay, now talking about the gynoecium which is there. Gynoecium is basically oh, superior in nature. It is what? Superior in nature. Okay, so that is why it is above the line. That means it is at the top. The gynoecium is at the top, yeah. And there are two of them, and two of them basically they are fused to each other. They would be fused to each other. So this is the thing that is there for Chili or Solanaceae family. Please do not forget. Very, very important thing. Cool, cool. Now going ahead to the next chapter, that is cell cycle and cell division. I request you to take a break. Okay, take a break, as in pause the video, go through all the questions once more and come back. Okay, do not go elsewhere, be here itself, because this is a very short time. It's just 20 minutes now. So just go through it once and come back again. We'll start off cell cycle and cell division. I'll be right here. Okay, just a second. Chalo, cell cycle and cell division. Oh, up over here we have question number 1. Identify the correct statement with regard to G1 phase of interphase. So you know, interphase is a phase where the cell takes break, and it tries to grow in size and also replicate its DNA, replicate the important organelles, because soon it is going to enter into what? The M phase, where the actual division takes place, my dear children. So over here, according in interphase also, I told you it's a very big phase where preparation happens, right? So it is segregated into few parts: G1 phase, S phase and G2 phase. Yeah, and you are asked what is correct regarding the G1 phase, the first phase which is there. So DNA synthesis or replication takes place over here. Wrong. So over here, my dear children, suppose this is the M phase where the actual division takes place. This whole thing is an interphase. Interphase is divided into 3 parts: G1, S phase and G2 phase. G1 phase is where most of the cell growth process and things like that takes place. Okay, what happens? The cell will be growing in size, they will be metabolically active and things like that. There will be certain organelles, only few organelles that will be replicating during this particular point of time. S phase or synthesis phase, this is where the DNA replication will happen. The DNA which is there, obviously when a cell divides into whatever content is there, it has to also divide into 2, right? So it has to duplicate. So DNA replication and also centrosome replication happens in S phase. G2 phase is where the other organelles replicate whatever were left behind. Only few organelles duplicated over this time, right? So they would be also duplicating in G2 phase, whatever are left behind, and also certain very important proteins would also be duplicating in G2 phase. Good, got it? Okay, so DNA synthesis or replication takes place over here. It will be a very wrong statement because it happens over here in S phase. Now, reorganization of all cell components takes place over here. Cell is metabolically active, grows but does not replicate its DNA. Yes, cell is metabolically active, they will be growing in size but is not going to replicate its DNA. Nuclear division takes place over here. No, my dear children, nuclear division would be taking in the M phase. Nuclear division, and then what? And then the cell division also will be taking in the M phase. So I'll say this is also wrong and this is also wrong. Okay, reorganization all happens later. So your answer for this question is what? This. This is the only correct statement regarding G1 phase or gap 1 phase of interphase. Now my dear children, I'll tell you one more thing. The cells that do not want to enter into the cycle, they exit from the G1 phase and they get into G0 phase. Okay, G0 phase, or you also call it quiescent stage and things like that. Resting phase, G0 phase and things like that. Okay, please remember that it is exiting from the cell cycle but after G1. Okay, so yes. Some dividing cells exit the cell cycle and enter the vegetative inactive stage. This is called quiescent stage. This process occurs at the end of what? M phase, that is the mitotic phase where the actual division takes place, G1 phase, S phase or G2 phase. It is at the end of G1 phase. I just told you. Example, your heart muscle cells, my dear children, they are metabolically, remember this is there, okay, very important point. The ones that are there in G0 stage, they are metabolically active. They are metabolically active. Do not forget that. So some dividing cells exit the cell cycle and enter vegetative inactive stage. This is known as quiescent stage. This process occurs the end of G1 phase. Cool, cool ma'am. Chalo, next. Given below is a schematic breakup of phases, stages of cell cycle. Which one of the following is the correct indication of stage phase in the cell cycle? So over here A, B, C is given and here D is given. So my children, okay, so over here mitosis is given. So this one which is there, it is interphase, right? And interphase, what all do you have? G1, this would be S and this would be G2 phase. right? So D matches with S. D matches with S. So I had just a one option like that, and that's how I got my answer also. But still, I have to recheck the other options. Okay, D is synthesis phase. Now going ahead, my dear children, I have A, B, and C. So what is A over here? A whole thing is known as karyokinesis, nuclear division. A whole thing is known as karyokinesis, that is nuclear division. Now the nuclear division is divided into 4 stages: prophase, metaphase, anaphase, and telophase. So B should be anaphase. So B which is there, it should be anaphase. What is A? A is karyokinesis, the division of the nucleus. Now after this, after B you have this gap, that is telophase, and after that the whole nuclear division has taken place. Now the cell has to divide, and that is represented by C, that is known as cytokinesis. So cell division is known as cytokinesis. So C is not karyokinesis, it should be cytokinesis. A which is there, it should be karyokinesis, not cytokinesis, and B which is there should be anaphase, not metaphase. So hence you got your answer, that is small b. That is D is equals to synthesis phase. That is the only correct thing that is given up over here. Okay, so a cell takes around 23 hours over here for preparation of the division, but takes only 1 hour for the actual division, that is this phase. Cool? Yes. Next, in the somatic cell cycle, in G1 phase, DNA content is double the amount of DNA present in the original cell. In G1 phase, DNA content is double the amount of DNA present in the original cells. Why? It will be the same, right? It should be the same. It's a somatic cell. It's not a gametic cell. In gametes, my dear children, what happens is that the number of chromosomes, or the amount of DNA which is there, it will be reduced. It will be reduced. That is why it is known as reductional division, right? So this is absolutely wrong when you talk about somatic cells. Wrong. Okay, now DNA replication takes place in S phase. Yes, DNA replication always takes place in S phase only. So this is correct. A short interphase is followed by long mitotic phase. Wrong. I told you 23 hours long interphase is there, not short interphase. It's long interphase. So this is wrong. G2 phase follows the mitotic phase. After the mitotic phase, you have G1 again the whole division. Not G2 does not follow the mitotic phase. It will be wrong. Okay. Fine. Cool. So what is your answer for this question? DNA replication takes place in S phase. Easy question. Came in 2004. Okay. Now which of the following options gives the correct sequence of events during mitosis? During mitosis. Okay. So condensation. Basically, I told you prophase, metaphase, anaphase, telophase is there. I'll write it down for you. So you have prophase, metaphase, anaphase, and then you have telophase. Agree? Okay. Now in prophase what happens? The chromatin fibers which are there, they condense to form chromosome. So condensation happens over here. Nuclear membrane disassembly. Basically the nucleus which is there, its membrane also goes off. So that also happens over here. Now the chromatin condenses to form chromosomes and things like that. What will happen? Now these chromosomes which are elsewhere in the cell, my dear children, they would be taken and put in one particular equator. Arrangement at the equator, that is the metaphasic plate. So this is known as, this is happening in the metaphase. Now the centromere division. What do you mean by centromere division? Basically it looks like this. So your chromosome is like this. Basically what happens is that they are arranged in the center. Now slowly spindle fibers would be coming and attaching over here, and it will be dividing this particular centromere, the point of attachment, into two. Now you would be having what? You would be having something like this. So division of centromere division took place, right? And then after that they will be segregating, they will be moving away, my dear children. This happens, okay? Centromere division as well as segregation would be happening in the case of anaphase. They would be moving around, and then towards the end you have the telophase, but they finally moved away, and the the nuclear membrane which went off, it will be coming back again. All the organelles which went off, it all will be coming back in the telophase. It's like the restart phase. Okay. So basically this is your answer. The first option itself is the answer. Condensation, crossing over and things like that. Remember, you're talking about mitosis. You have two types of cell division, mitosis and meiosis. Mitosis happens generally in the somatic cells. Meiosis happens in gametic cells because you need reduction there. And in gametic cells, you have the new recombinations or basically chromosomes crossing over and things like that. In the case of mitosis, you do not have all this thing. So crossing over is there, wrong. Okay, arrangement, condensation, the arrangement equator, then centromere division, segregation, telophase. A lot of things are still missing. So I'll say this is the correct answer, right? Condensation, nuclear membrane assembly, crossing over is not there. I told you already. So what is your answer? Your answer is A. Cool. Next. Spindle fibers attach on to. So ma'am, you just told spindle fibers basically are going to come and attach over here on the centromere. No baba. Centromere division takes place. I told you they segregate also. But where are they attaching? That I didn't tell you. Okay, so centromere basically attaches at the kinetochore. Where are they going to attach? They're going to attach, not the centromere, sorry. Spindle fibers are going to attach on the kinetochore. That is over here. Right over here. Always remember. So spindle fibers attach onto the centromere of chromosome wrong, kinetosome of chromosome wrong, telomere of the chromosome wrong, kinetochore of the chromosome correct. This is your answer. Okay, it attaches to the kinetochore of the chromosome. Now going ahead. A stage in cell division is shown in the figure. Select the answer which gives correct identification of the stage with its characteristics. So cytokinesis, telophase, late anaphase is there. So what is this stage, my dear children? Well, basically the chromosomes have moved away. You can clearly see that there is no chromosome represented over here. Instead, there is nuclear membrane being formed and stuff like that. There is a whole nucleus and stuff being formed. Okay, so where do you find it? In telophase, where everything what reappears, my dear children. And you can see that the cell also have started division. Okay, there is a cell furrow being formed over here. So this whole thing, okay, so it's like telophase. It's like telophase where basically what is happening? My dear children, they have segregated on the opposite poles, yeah, and now slowly things are being settled down. The next process would be cytokinesis. So over here they have said this is cytokinesis, cell plate formed. My dear children, it would have been cytokinesis. I would have thought about that, but over here it is said cell plate is formed. Cell plate is basically, you should be having something like this. In the middle you should be having something like this, and this should be moving out. That is not cell plate formation that happens in the case of plants. Okay, in the case of animals, cell furrow formation takes place over here. There is a furrow being formed over here. So this itself is wrong. So I cannot consider cytokinesis over here, even though I could have considered if there was cell furrow formation mentioned over here. Now telophase, endoplasmic reticulum and nucleus not reformed yet. Wrong. In telophase, my dear children, if it's assume that I am saying it's telophase for sure. But this statement is wrong. Endoplasmic reticulum and nucleus not reformed yet. It's wrong because in telophase everything will be reforming back. So this is wrong. Telophase, nuclear envelope reforms, Golgi complex, they will also reform. Yes, in telophase that would be happening, and this is exactly telophase, yeah. Late anaphase, chromosomes move away from the equatorial plate, Golgi complex not present. My dear children, it cannot be late anaphase. You should be showing what chromosomes up there. There is no scene of chromosome up over here. So I would not say it is late anaphase or not. Okay, this would be just this. Cool. And towards late anaphase, it's not they're moving away from the equator. They've almost reached, almost. Okay, they are moving away, but they would be almost reaching towards the poles. Now, if you are provided with root tips of onion in your class and are asked to count the chromosomes, which of the following stages can you most conveniently look into? So in the root tip experiment, you might have all done it, right? So which is the stage that you usually see? Metaphase, telophase, anaphase, prophase. The most easiest one to spot the chromosome, it is metaphase, where all of them, they are in the center, they are lying together at the equatorial plane. So your answer for this question would be metaphase. A is the answer. Now, going ahead. Match the following with respect to meiosis: zygote, picote, diploid, dikinesis. So listen to me carefully, it will be really helpful for solving all questions coming off from this topic, okay. So lazy zebras play during day, okay. So leptotene is basically where the chromatin again start condensation. They would be starting condensation, okay. First of all, let me tell you what I am talking about. So basically all the steps comes under prophase. Remember prophase, metaphase, anaphase, telophase, yeah. So when meiosis happens, my dear children, you have 2 rounds of all these things. I mean not all this thing, prophase, metaphase, anaphase and telophase. 2 rounds as well. There will be prophase 1 and there will be prophase 2 also later on. In prophase 2 you do not have this segregation. Only in prophase 1 you will be having this segregation. So I will say meiosis, it includes P1, P1, M1, A1 and T1. And then in the case of meiosis 2, there is second cycle of meiosis, so it will be P2, M2 and A2 and T2. Fine. This is how the segregation usually works. Fine. Now, my dear children, you have to listen to me carefully. I am talking about prophase 1. What happens in prophase 1? That is a question. So, basically DNA replication, everything happened initially, okay, initially in the interphase, right? Now, we have got into the M phase. Now, in the M phase, in the leptotin stage, the chromatin which is there, it will start condensing to form the chromosome. Also, the nuclear membrane, everything will be starting to disappear. That is a normal thing that happens in leptotin. Zygotin, what happens over here? So, two chromosomes would be coming together. What chromosomes, ma'am? The homologous chromosome. What do you mean that by, ma'am? So basically, you know, we get one chromosome each from both of our parents. Okay, for example, this is first pair. We have 23 pair of chromosome. First pair, we got one from our father, one from our mother. Right? So we have a chromosome like this, a chromosome like this. So in the S phase, initially, before all this, in the interphase which was there was S phase. In the S phase, what happened? Replication of DNA happened. So it looks like this now. It looks like this. There is a replication of this also. So what do you call these two strands as? You call them sister chromatids. You call them sister chromatids. What do you call them? These two strands, they are non-sister chromatids. They are non-sister chromatids. Now what are these two things which you got? Which you got from both of your parents, the first pair? You call them homologous chromosome. Why, ma'am? Why homologous? Basically, over here, whatever gene is there, okay, this gene would be here also. As in, different variants can be there of a gene. This gene codes for height, this gene also codes for height. This can code for tall height, this can code for dwarf height, but anyway, it is just height, right? So, at the same position, you have a gene coding for height. So, those genes, okay, or any character, my dear children, which should be similar, they are known as homologous chromosome. Right. So, what happens is that in the case of zygotin, the homologous chromosome would be coming together. Okay, the homologous chromosome which is there, it would be coming together and forming what? And they would be glued up by something, that is, glued up by something, and together, now this will be known as synaptonymal complex. It will be known as synaptonymal complex, my dear children. Cool? Works? Now, going ahead. So, synaptonymal complex is being formed. This process is known as synapsis. This process would be known as synapsis. Now, going ahead, you have pi chitin. What happens in pi chitin, my dear children? In pi chitin, the non-sister chromatids which are there, okay, this and this, okay, this and this would be crossing over. They would be exchanging their stuffs over here. So, basically, it will be looking like this. So, they are going to exchange the stuff. This process that is happening over here, this is known as crossing over. And this happens only with one enzyme that is known as recombinase. Recombinase, that will be helping in the formation of recombination, new combinations up there. Okay, then you have dikinases. What exactly happens in dikinases? Basically, these guys, the synaptonymal complex is also there over here. Remember? Synaptonymal complex will be dissoluting. It will be going off. It will be going off. And when it goes off, and basically, the chromosomes are still attached together. Okay, at one particular point after dissolution, the chromosomes are still attached together at one particular point, and this is known as chiasmata. This is known as chiasmata. Now what will happen? This is known as leptotin, then zygotin, then pi chitin, and then diplotin, and then dikinases. In dikinases, basically, when they try to break up really hard, my dear children, what will happen? This chiasmata which is there, it will move towards the terminal position. It will move towards the terminal position. So, I will call this terminalization of chiasmata. I will call this terminalization of chiasmata, my dear children. So, match the following with respect to meiosis. Now, I hope you will be able to do that. Okay, what happens in zygotin? In zygotin, my dear children, you have synapses being formed. So, A goes with 4. A goes with 4. I just have one option, and with that I got the whole answer. But let's still go ahead. In pi chitin stage, what happened, my dear children? Crossing over happened. In diplotin stage, what happened? The synaptonymal complex went off, but still they are stuck together in a cross-section like view. What is this? This is known as chiasmata. Okay, diplotin stage. Now, dikinases, terminalization. So, hence, this becomes your answer. So, lazy zebra play during day. Remember like that. It will be really, really important for you. Now, crossing over takes place between which chromatids and which stage of cell cycle? I told you. Crossing over happens between which chromatids? Non-sister chromatids, agree? Of what chromosome? Homologous or non-homologous? Homologous chromosome. So, non-sister chromatids of homologous chromosome. And in which stage, my dear, crossing over takes place in zygotine stage, right? Lazy zebra play. Is it zygotine or picotine? It's picotine stage, okay, it's picotine stage. So, non-sister chromatids correct, non-homologous chromosome wrong, zygotine wrong. Non-sister chromatids correct, okay, homologous chromosome correct, picotine stage of prophase 1, that is also correct. Now, to confuse you guys, they can be giving you prophase 2 also. Do not, please read the question very carefully and then go ahead, okay. So, it is prophase 1 where all this thing happens. Now, non-sister chromatids of homologous chromosome at zygotine stage of prophase 1, wrong. Non-sister chromatids of non-homologous chromosome at picotine stage of prophase 1, no. Your only answer is B. Fine. Going ahead, match the stages of meiosis in column 1 to the characteristic feature in column 2 and select the correct option using the codes given below. You have picotine, metaphase 1, dikinesis and zygotine. So, what happens in picotine, my dear? Lazy zebra play, crossing over, takes place over here. In metaphase 1, and all the metaphases, my dear chromosomes would be would be arranging themselves in the equatorial plane or the metaphasic plate. So, I will say chromosome align at the equatorial plate. Dikinesis, at the end of prophase 1, you have dikinesis, terminalization happens, right. Now, zygotine, pairing of homologous chromosome. What is the answer for this? A goes with 3, A goes with 3, I just have one option, which is why this becomes my whole answer. B goes with 4. Okay, C goes with 2 and D goes with 1. Cool? Yes. Next, the enzyme recombinase is required at which stage of meiosis? Enzyme recombinase, at which stage do you require that? Recombinase is for recombination, that means crossing over. And where does crossing over happen? In the picotine stage. As so many questions are coming from the same topic but in different manner, which is why I am discussing it right now. Okay, so 12 goes with A. Now, question number 13. The given figure is a representation of certain event of a particular stage of a type of cell division. Which is this stage? Which is this stage, my dear? If you see, the crossing over has taken place, they have exchanged certain things, right? So, which is this stage where all these things have been completed? Prophase 1 during meiosis, prophase 2 during meiosis, prophase of mitosis, both prophase and metaphase of mitosis. My dear children, where do you see this? Where do you see this? You would be seeing this in prophase 1 during meiosis, okay? After the end of prophase 1, you will be seeing something like this. Not during prophase 2. Okay, not during prophase 2. In prophase 2, these guys also gonna separate out into two separate things. So, it will be looking something like this and this, where one would be having some new thing up over here like this. Okay, so that they also separate in prophase 2. You will be knowing that, yeah. Now, prophase of mitosis and mitosis, this all does not happen. Both prophase and metaphase of mitosis wrong. Only this is the answer. Cool. Now, when paternal maternal chromosomes change the materials with each other in cell division, this event is known as paternal maternal exchanging their stuff in homologous chromosome. What do you call this? Do you call this bivalent forming? Bivalent forming means, yeah, both of them are basically coming together. That's all. This is bivalent formation where both of them are going to come together. Tetrad, diad forming means basically two chromosomes, two chromatids coming together. Synapses means you know already, and crossing over, my dear, and this is the actual answer. This is where the change of the materials happens between each other, that is the maternal and the paternal chromosome. So, this is your answer, D, crossing over. Cool? Yes. Going ahead. Meiosis 1 is reductional division. Meiosis 2 is equational division. Due to what? Meiosis 1 is reductional division. Why? Why? Because basically over there, if you are starting up with 46 number of chromosome, okay, in meiosis 1, my dear children, it will divide. At the end of meiosis 1, you get two cells each with 23 chromosomes each. Yeah. Again, you have meiosis 2 that is happening, where the cell will again divide into 2. Do you think again halving will happen? No, it's just 23. So, it has reduced from 46 to 23 over here, which is why it is known as reductional division. it is known as reductional. So meiosis 1 is reductional division, meiosis 2 is equational division. Why it is known as equational? Meiosis 2 is equational, my dear children, because over here you saw this divides into 2. Again you get 23, 23 only. Is this reducing? No. So this is like equational division. This is like equational division. Let me tell you, in total meiosis is considered to be reductional. If you're talking about meiosis 2, it is just like mitosis where the same number of things are there. So it is known as equational division. So pairing of homologous chromosomes happens in meiosis 1, crossing over happens in meiosis 1, disjunction of homologous chromosome, that means separation of homologous chromosome, that also happens in what? In meiosis 1. Separation of chromatids. Separation of chromatids. That means I told you initially over here, these are the number of chromosomes that we have. Even during metaphase, they come over here like this and they get separated. It's like the homologous chromosomes are gonna separate. When you talk about an homologous chromosome are gonna separate, so one cell is going to receive this and the other cell is going to receive this. Agree? Agree? Now, in the case of the second phase, what happens? Basically, they too separate. The chromatids are gonna separate over here. The chromatids are going to separate. When you separate chromatids, what's the good thing about that? Basically, you know, this whole thing is known as one chromosome. This whole thing is known as a second chromosome. Because both of the chromatids are attached with the help of one centromere. So I do not count it as 1, 2, 3, 4 chromosome. I'll just count it as one chromosome and the second chromosome. Right? But in the case, now they have separated. In the case of meiosis 2, what is happening? This particular chromosome is also going to divide into 2. So I'm going to get 2 chromosomes out of it. Out of 1, I'm going to get 2 chromosomes, which is why the number equation remains the same only. The equation remains the same. 23 here, 23 here. I hope you got it. I hope you got it. So answer is separation of chromatids. The chromatids get separated in meiosis 2, which is why it is equational division. Cool. Chalo. Next thing, we have body fluids and circulation. My dear children, if you have not understood this question, do let me know in the comment section. I know it's kind of confusing. Okay, I'll try to make you understand. Okay, with one last attempt. So over here, I have two chromosomes. Agreed? 2 chromosomes. Because both of them are having a common centromere, I cannot count this as 1, 2, 3, 4. I'll count this as 1 and this as 2. Right? By the end of meiosis 1, both of them separate. Okay, both of them separate. I'll say here it had 2, but here it will be having 1 chromosome and 1 chromosome. 46 was there, it became half, 23 and 23. 2 ka half, 1 and 1, right? Now in meiosis 2, what will happen? This particular chromosome which is there, it is dividing into 2. One chromatid is going to go over here, another chromatid is going to go over here. So the cells which arose from this cell are still having one number of chromosomes. Similarly over here also, when it divides into 2, this goes here, this goes here. Since this chromatid separation took place, that is why I've got 1 and 1. Otherwise, I would not have got 1. So this was 46. This became 23, 23, and now this also is 23, 23 because of separation of chromatids. I hope you got it now. Yes, yes. Now we have the next chapter, that is body fluids and circulation. Let's get into it. So, match the following columns and select the correct option. Column 1 and this is column 2. Eosinophils, basophils, neutrophils and lymphocytes are given. What does eosinophils do? Eosinophils are actually produced during allergies. And what do? They actually try to moderate the allergy. How? What is that something that causes allergy at the first place? Histamine and things like that. Yeah, there is histamine production that is happening in our body. So, there should be someone who should be breaking down the histamine, and that is done by eosinophils because they are going to release histaminase. Okay, and also certain destructive enzymes which are there, which is going to disrupt what? The histamine and all the other components which are there. So I say A goes with 3, A goes with 3. I have just one option and this should be my answer. But let's go ahead. Basophils. Basophils are the ones that releases histamines. Okay, they do the opposite of it. Basophils are the one that causes inflammation. Inflammation is what? Basically when your blood vessels and everything get what? Dilated, so that all the cells can come and rush in and help in whatever thing is happening. That is the actual thing for basophils. It does a good work, but basically the consequences of it is what? Inflammation and things like that. So when it does, there is inflammation being seen. And over here in the case of basophils, my dear children, they would be having, they are very peculiar, they look different because they have so many granules which would be releasing what? Lot of histamine. So basophils goes with 4. Release granules containing what? Histamine. Cool. Now neutrophils are there. Neutrophils are the most abundant WBCs out there. Remember always, neutrophils are the most abundant and basophils are the most least. So neutrophils are the one that does phagocytosis. They take in the cells, okay, whatever component is there which is not needed, they'll take it in and kill it inside their body, inside the cell body. Okay. So neutrophils goes with phagocytosis. We have lymphocytes left. D. Lymphocytes are two type. They produce B lymphocytes, or basically you have T lymphocytes. Both of them are very important in immune response. So D goes with 1. D will be going with 1. So A with 3, B with 4, C with 2, and D with 1. Cool. This becomes your answer. Next, match the items given in column 1 with those. I think I numbered question, no. Okay, match the items given in column 1 with those given in column 2 and select the correct option given below. Fibrinogen, globulin, albumin. So all of them are found in our blood, my dear children. Yes, fibrinogen, they are very much important for blood clotting. They would be converting into fibrin and things like that, active fibrin and things like that. So they are important for blood clotting. Globulins, immunoglobulins, IgA, IgG, IgE, you know, they are immunoglobulins. Okay, apart from immunoglobulins, you also have other things. Yeah, basically all of them, the antibodies which are being produced, what do? They are a major thing for defense mechanism. Right. Now what you're left with? You're left with albumin protein. What does it do? Albumin protein is responsible for water retention and things like that. Okay, it will be taking in certain things, okay, so that the water retains. So can I say it is responsible for osmotic balance? I can say that. So basically A goes with 2, okay, over here. B goes with 3, okay, and C will be going with 1. So this becomes your answer. Now going ahead, adult human RBCs are enucleate. Which of the following statements is or are most appropriate explanation for this particular feature? Enucleate means they do not have nucleus, my dear children. Okay, adult human RBCs, they do not have nucleus. Why is that so? First thing, I'll just say abruptly, okay, the thing is, when a cell are forced to lose its nucleus, probably it is doing very important function, and for RBC, you know the important function, it is the one that carries what? Hemoglobin, and hemoglobin is the one that carries oxygen. So basically for having internal space available for oxygen transport, my dear children, you would be having what? RBCs as enucleate. They would not be having the nucleus. So they do not need to reproduce. Yes, actually RBC do not reproduce, that is also there, but that is not the reason for this. They are somatic cells, considering they specialized cells, actually they carry oxygen. So but yeah, they do not metabolize. They do metabolize, that's not rare, but the amount of metabolism would be less, but yes, they do. All their internal space is available for oxygen transport. This is their answer. This is the actual answer. So your answer would be D, only 4. Cool. Now next, person with blood group AB is considered as universal recipient because he has. So person with AB blood group, so A and B cells are there. What do you mean by A and B? So basically they would be having A antigen at the top and it will be having B antigen at the top. Suppose in their blood plasma, if there is anti-A and anti-B antibodies being produced, both the cells would be killed. So that is why they would not be having antibodies against any antigens. So which is why they are considered to be universal recipient. They can receive either A blood group, they can receive B blood group also, because whatever comes in, okay, suppose A is coming, that means they would have A antigen. Suppose B is coming, that will have B antigen. But to fight back against it, do they have antibodies? No. Which is why I'll say they are universal recipient. They will be taking in certain things. Okay, so what is the universal donor? Can anyone tell? Again, I want to see that vigor in you. Yeah, can anyone tell me which one is the universal donor and why? So, person with blood group AB is considered as universal recipient because he has both A and B antigens on RBC but no antibodies in the plasma. Correct. They have antigen on the surface but no antibodies in the plasma. Both A and B antibodies in the plasma wrong. No antigen on the RBC, no antibody in the plasma. Both A and B antigens in the plasma but no antibodies wrong. Now, going ahead. The figure shows a human blood cell. Identify it and give its character. You have this. What is this showing? This is a basophil, my dear children, and basophil would be having big granules out there that would be containing the histamine and the other things, okay, serotonin, histamine and things like that. So, the answer is basophil secretes serotonin inflammatory response. It's not B lymphocyte, neutrophil or monocyte because all of them would be having a peculiar design in their nucleus. The nucleus itself is not visible over here. They definitely have a nucleus. Don't think they do not have it. They definitely have a nucleus, but that is outshined by the number of granules up over here. So, your answer would be A, basophils. Next thing, a person with unknown blood group under ABO system has suffered much blood loss in an accident and needs immediate blood transfusion. His friend who has valid certificate of his own blood type offers the blood donation without delay. What would have been the type of blood group of the donor friend? So, it is asking about a universal donor. That means anyone who can donate, because here you are not told about the blood group that the patient has, right? So, basically what is that something that can be given to all? Not now, actually in today's generation we did not put it like that, but yes. So, it is type B, type AB, type A, type O. Your answer is type O. Why type O? Because my dear children, in type O blood group you definitely have antibodies. You definitely have antibodies, but do you have antigens on the surface? No, it does not have either A or B antigens, which is why it is known as O blood group. There is nothing. There is nothing over there. So, if I put the cell inside, okay, basically I am going to put the cell inside another plasma, right? So, basically when I put the cell inside, what is going to happen is that the blood cells which are there, they are not going to ignite any response out there because they do not have the antigens. So, even if that person is A blood group, B blood group, whatever antibody they produce, there is no antigens to, there is no antigen on this surface to attack on it. So, which is why type O is known as the universal donor. Cool. Next. The most popularly known blood grouping is the ABO blood grouping. It is named as ABO and not as ABC because O is referring to having what? O dominance of this type of genes for A and B types, no. One antibody only, either anti-A or anti-B on the RBCs. Okay. No antigens A and B on the RBCs or other antigens beside A and B on the RBCs. What is it? They do not have any antigens, not A nor B, which is why it is known as O blood group. Next, erythroblastosis fetalis is caused when the fertilization takes place between the gametes of what? Rh negative female and Rh positive male. So, there is Rh antigen, my dear children. Some cells would be having this Rh antigen, they are known as Rh positive people. Some cells we do not have this, we call this Rh negative, okay. So, that is the whole thing. Yes, just a second children. Yes. So, yeah, so those who do not have it, it will be Rh negative, right. So, basically what happens, erythroblastosis fetalis is when the mother is not having the antigen, which is why she will be Rh negative. That means negative blood grouping is there. Now, the baby, suppose, okay, the baby would be having positive blood group. In that sense, what will happen? Basically, there is a baby with cells that has the antigen, and suppose the mother's and the baby's blood becomes transfused, okay, become exchanged. What will happen? The baby's blood, okay, the baby's blood, since it has antigen on the cell surface, right, what will happen? The mother would be producing antibodies against it and the baby would be killed. Do you need that? No. Even it will be very harmful for the mother as well. So, that is a thing, my dear children. So, basically where do you see it? You would be seeing it when the mother is Rh negative and the father is Rh positive. That is when the child can also be an Rh positive person, okay. So, that is a thing. So, Rh negative female and Rh positive male, that is your answer for this particular question. Rh positive female and Rh negative male, no. Mother do not have it, that is why she will be producing antibodies against it. You get it? So, mother has to be negative and the child which will be there, it will be positive. So, the father also should be positive, then only the child will be getting it. Next, carbonic anhydrase occurs in lymphocytes, blood plasma, RBCs or leukocytes. What do you think carbonic anhydrase does? Carbonic anhydrase is something that would be converting the CO2 which is there into bicarbonate ions. H2CO3 and things like that. That is H2CO3 first and then HCO3, that is the bicarbonate ions. So carbonic anhydrase is responsible for that, and that is something that you would be finding in RBCs, not anywhere else, not in the lymphocytes, not in the blood plasma, only in the RBCs. Yeah. Next, which one of the following is correct? Lymph is equals to plasma plus RBC plus WBC. Wrong. Lymph is basically when the capillaries, they leak out. Okay, when they leak out, my dear children, capillaries, they have small pores, right? And with the pressure that blood comes, there will be some leakage happening. It will be obviously containing plasma. It will be containing WBC. Even though WBC are big, they can squeeze into and move out. That is why WBC will be there. RBC is not going to be there in the lymph. Always remember that. Blood consists of plasma, RBC, WBC, platelets. Yes, plasma plus all the cells which are there together, it forms the blood. This is correct. Let's see the next thing. Plasma is equals to blood minus lymphocytes. Plasma is equals to blood minus lymphocytes, my dear children. It's not just lymphocytes. It should be all the other cells, that RBC, WBC and platelets. Lymphocytes just a part of WBC, but all the other WBC are also there, right? So it cannot be blood minus this. It should be blood minus these things which is mentioned over here. Serum is equals to blood plus fibrinogen. Wrong. Serum is equals to what? Plasma minus the clotting factors and stuff like that. Plasma which is there, the liquid part which is there, minus the clotting factors which are dissolved in it. That is what you call as serum. So basically this is the only correct answer that we have. Next, the lymph serves to return the interstitial fluid to the blood, return the WBCs and RBCs to the lymph nodes, transport CO to the lungs, transport O2 to the brain. The lymph serves what? I told you lymph is basically a collection of all the extra fluid which leaks out, yeah, and this fluid has to be given back to the blood vessels. Why? Basically, otherwise if you're not providing it back, what happens is that the blood pressure is not going to be maintained. So lymph serves to return the interstitial fluid to these blood. That is the answer. Interstitial fluid as in between the cells, my dear children, the blood vessels are going to leak out something, yeah. The blood vessels are going to leak out certain things, and this fluid which is there in between the cells, you call this interstitial fluid. Interstitial fluid, because it is between the cells, interstitial cells. So basically the lymph serves to return the interstitial fluid to the blood, return the WBC and RBC to the lymph nodes, transport CO2 to the lungs, transport O2 to the brain. What is the answer? Return the interstitial fluid to the blood. This fluid has to go back to the blood. That's how pressure and everything would be maintained. Got it? Yes. Next, match the column 1 with column 2. Column 1 is P, QRS wave, T and reduction in the size of T wave. Listen, P wave is there, was the first wave which is there. It shows the contraction of both the atria. You also call this as depolarization of atria. What do you call this? Depolarization of atria. Okay, now when they relax, you call them repolarization. So depolarization of atria occurs during the P wave. Okay, so P wave goes with 4. P wave goes with 4, so I have two options like that. I can cut off these two options. Going ahead, QRS complex. After P, you have QRS. So atrium contracted, it sent down the blood to the ventricles. Now it's time for the ventricles to contract, and that will be known as depolarization of ventricles. So can I say B goes with 1? B goes with 1, my dear children? Yes, I can say that. Now what about T wave? At the end there is T wave, but what is happening? Ventricles have contracted, now they stay slowly relax. They will be slowly relaxing. This is known as repolarization of ventricles. Right. Now suppose there is a reduction in the size of the T wave. The T wave which is there is a reduction in the size of that. What does it show? It shows coronary ischemia. That means that basically the heart muscle cells which are there, they are not getting enough amount of oxygen. That is what it means by this, because of probably debris formation in the blood vessels and things. Okay, so P means contraction of atrium. Okay, QRS means contraction of ventricles. At the same time when the ventricles are contracting, the atrium which is there, it will be relaxing. So QRS is showing what? Depolarization of ventricles, and also one more thing, that is repolarization of atria. That means atrium is going to relax. Always remember that. Okay, and T wave means repolarization of ventricles only. Fine. Now match the items in column 1 with those in column 2 and select the correct option. Tricuspid wall, bicuspid wall, semilunar walls. So where do you find tricuspid wall? Suppose this is the atrium, right atrium, left atrium, right ventricle, left ventricle. Okay. In between the right atrium and the right ventricle, you have the wall, and this valve is known as tricuspid valve. You can remember like that. There is a valve up over here. This is known as bicuspid valve. Okay. Now, this is going to open into aorta, and this is going to open into pulmonary artery. There is a valve up over here. These valves are known as semilunar valves. Semilunar valves. Why? Because they are half moon shaped. They are what? Half moon shaped. That is why it is known as semilunar valves. So tricuspid valve, my dear children, it is between the right atrium and right ventricle. Bicuspid valve, it is between left atrium and left ventricle. Semilunar valve is between the right ventricle and the pulmonary artery, or it can be between left ventricle and the aortic artery, aorta. Okay, so it is known as aortic valve, and this is known as pulmonic valve as well. Please remember that. So basically, A goes with 3. You just have one option like that. B goes with 1, and this. Next thing, the diagram given here is a standard ECG of a normal person. The P wave represents what? What does the P wave represent? What does the Q represent? What does this represent? I already told you. P wave represent what? Contraction of both the atria. What do you also call it? You also call this as depolarization of atria. Okay, or atrial systole, or you also call this atrial systole. So beginning of the systole, what systole? You didn't mention what systole, atrial systole, diastole, or whatever thing is there. Okay, end of the systole, contraction of both the atrium. You have mentioned it's about atrium. Yes, that happens over here, and this is also known as, what do you call this? Depolarization. So this is correct. Initiation of the ventricular contraction, no, that happens over here. Ventricles contract up over here. So initiation happens in the Q, not there. Okay. Now, which one of the following has an open circulatory system? So it's a mix of animal kingdom and this particular chapter. Open circulatory system means there is no designed blood vessels and up over there. Okay, they definitely have heart and stuff. Okay, where do you find it? In arthropods and most of the mollusk. Okay, you have arthropods and most of the mollusk that has what? That has open circulatory system. Rest all of them have closed circulatory system. So over here I can spot one mollusk, that is octopus, but octopus has a more complex circulatory system, so I would not count it as an open circulatory system. Yeah, Ferritema is basically coming under annelids. Yeah, Hirudinaria is also coming under annelids, that is leech, right? Now Periplaneta, that is what? Cockroach, and cockroach is what? Arthropod, and all the arthropods, they have open circulatory system. So this becomes your answer. Okay. Next, the correct route through which pulse making impulse travels in the heart is. So basically, again, I have to make the heart. Right atrium, left ventricle, right ventricle, right? So over here, the top you have a node that is known as SA node. What do you call this? This is known as SA node. Sinoatrial node, yeah? Sinoatrial node would be sending down signals to all the muscle cells out there. Yeah, it will be sending signal to right up over here. Here what do we have? We have AV node. Okay, AV node will say, hey relax, and then it will send down the signals to here. What is this? This is the bundle of His. This is the bundle of His. From here, the signal would be traveling throughout, and over here you have certain fibers which are known as Purkinje fibers. Okay, so basically SA node, then AV node, then bundle of His. Yeah, and then Purkinje fibers. When the signals are sent, first atrial contraction happens over here, then ventricular contraction would be happening over here. So SA node, then AV node, then bundle of His, then Purkinje fibers, then heart muscles. This would be your answer. Over here it is written Purkinje fibers first, so it cannot be. AV node first cannot be. AV node first cannot be. So the only answer that is here is B. Now going ahead, the hepatic portal vein drains blood to liver from where? So hepatic portal vein is a different type of vein. Why? Because usually children, what happens, there is an artery. The artery would be opening down into arterioles into capillaries, and from here you have veins, venules and capillaries. That is how things usually works. But in the case of hepatic portal vein, there is some difference. What is that difference? Basically, in the intestine, the vein would be carrying the deoxygenated blood back, right? But when it goes back, it should be directly going into the heart, right? But rather than, it takes a pit stop in the liver. Why? Because this vein which is there I agree it is deoxygenated, but it also has enough amount of nutrients, glucose and everything that is absorbed in the intestine. So this glucose and everything has to be transported to the liver. Why? Because over here in the liver, the glucose which is there, it is converted to glycogen and stuff. So the vein that comes in would be forming a capillary sort of system over here. Okay, and again it will be forming just a vein itself. It will be forming a vein itself because it has formed a vein already. It cannot make an artery up here. In that case, the artery would not be taking the blood to the heart, right? So it has to be a vein which will be taking this blood directly to the heart because it has deoxygenated blood. The only important nutrients which are there, it has to be transferred over here, which is why you have capillary being formed. So this is the hepatic portal system, and then again form a vein and then it goes away carrying the deoxygenated things. So your answer for this is intestine, because intestine is where maximum reabsorption of important nutrients happens. Cool? Cool. Not stomach, not kidney, not heart. Kidney is participating in renal portal system. There is another portal system between kidney and the lower limbs. Now, which one of the following statement is correct regarding blood pressure? 130 by 90 mm of Hg is considered high and requires treatment. What is a normal one? 120 by 80. So 130 by 90 is considered high, okay, fine, but does not require treatment. Sometimes it can be because of the activities that we are probably doing. So it does not necessarily require treatment until and unless it is very frequent. It is like this only, and it probably increases way too much. Yeah, so I'll say it is not entirely correct. 100 by 55 mcg is considered an ideal blood pressure. No, never. This is low. Diastolic pressure is really low. Okay. Now 105 by 50 mg makes one very active. No. 190 by 110 mm Hg may harm vital organs like brain and kidney. Yes, it will definitely harm brain and kidney because that is kind of really high pressure. We have 120 that is normal systolic pressure and 80 that is normal diastolic pressure, my dear children. Systolic means pressure when the ventricles are contracting. So pressure of the blood that it exerts on the walls when the ventricles are contracting. Obviously it will be higher than diastole. Diastole is basically the pressure that the blood extends on the blood vessels when it is in relaxed state, when the heart is in relaxed state. So that is fine. But if it's going really high to 190 by 110, my dear children, obviously it is going to harm the kidneys as well as the brain. Okay, because there should be an optimal supply of blood and everything. If you're doing this, definitely a lot of big problem. So this would be the correct statement out of this. Yes. And with this, we have completed 3 chapters, and also I'll definitely recommend you go solve as many questions as you can. Very, very important. It is very, very important for you to practice enough number of questions. If you're going there just assuming that I have studied the content and not practicing questions, it won't make any sense, my dear children. Question gives you a reality check, and I'll definitely recommend you to practice even more questions. Yes, so this is just a small effort from us with the limited time that is there, but still I'll recommend you, and as I told you, please take the mock test. It is definitely going to be helping you really well. And if you have not subscribed to the channel yet, do not forget to subscribe to our little channel as well. Bye bye. Take care.